So we just completed a first phase of a collaboration with Dr. David Tzagin, who's a pediatric endocrinologist at Hadassah Mount Scopus. And he identified a large family where some of the girls have a condition called ovarian dysgenesis. It means that even though they are female and they are girls, they don't, their ovaries don't develop, and as a result they don't go into puberty, and ultimately they don't have ovaries or eggs. And because this was a large family, we thought it was an opportunity to identify the genetic basis, or one of the genetic bases for ovarian development. And the reason that's important is because although there's a fair amount known about male sexual development, there's actually quite little known about what creates ovaries, and obviously this has great importance for issues like fertility later on. So we started studying the family. Basically, usually the way these studies progress is we compare the genetic makeup of affected women in the family with unaffected women and try to see what's different between those who are affected and those who are not. And we found such a gene. An important part of what we did was actually that we used really the latest technology in genetic analysis, which is called next generation sequencing. Now everything is next generation. Scientific name for this is massively parallel sequencing, where you can look at the entire human genome basically in one experiment. This is the first time that this has been done in Israel completely by an Israeli team. The sequencing itself was done at the Hebrew University, where they have next generation sequencer, and the analysis was done by us in collaboration with Liran Carmel's group from Hebrew University. And basically we identified the mutation that underlies this condition in these girls. The importance of this finding, first of all, for the family itself, now they can get tested. Often in these families, this is an Arab family, they have a high rate of marriage between relatives, so now relatives can be tested before they get married, so they won't have affected girls. Those who are already married, if we find they are at risk, we can then do pregestational diagnosis for them. So it's clinically important for the family, but it also has a lot of research implications and clinical implications, because this could be a gene that contributes to both male and female infertility in general, which is something we're going to look at now. And it's also an interesting tie-in between two processes that are usually thought to be different. It turns out that this gene is important both for creating sex cells, for creating ovaries and sperm, because it's important for meiosis. That's the division where cells, usual cells, normal cells, become sex cells that have only half the genetic complement. The other half of our genetic makeup will come from the other sex cell. And the other thing is that it turns out this gene is important for hormonal response, especially estrogen response. So it ties in these two processes of forming germ cells on the one hand and the hormonal response on the other hand. This particular mutation harms the ability to activate estrogen-related signals, which explains why it affects girls, but theoretically mutations which will affect signaling through testosterone might affect fertility in boys, and that's really going to be one of our next steps. And I think this collaboration, first of all, shows the power of collaborating between clinicians and people with the genetic laboratory experience and expertise, which is what we have here in the Genetics Institute, and also in our collaborations with Hebrew University. And it really shows the power of the new genetic technology that really allows us now to identify the basis for many diseases which are so common in our population.